There are moments in history when humanity takes a step forward so significant that the world is never the same again. The discovery of fire was one of those moments. The invention of the printing press was another. The arrival of electricity transformed civilization forever. And hidden among those revolutionary breakthroughs is a discovery that quietly changed every field of modern science: the discovery of the structure of DNA. Today, we live in a world shaped by genetics. Doctors can identify inherited diseases before symptoms appear. Investigators can solve crimes using traces of biological evidence. Scientists can study the origins of humanity by examining genetic markers passed down across thousands of years. Yet few people stop to ask a simple question: how did humanity first uncover the code of life itself? The answer leads to one of the most fascinating and controversial stories in scientific history. It is a story of ambition, competition, brilliance, and a woman whose contribution was almost lost to time. Her name was Rosalind Franklin. To understand why her story matters, we must first understand the scientific world she entered. During the first half of the 20th century, biology was undergoing a revolution. Scientists had already learned that traits could be inherited from one generation to another, but nobody fully understood how the process worked. The existence of genes had been proposed decades earlier, yet genes remained mysterious. Researchers knew they existed, but they could not see them. They could not explain how they carried information. They could not explain how a tiny fertilized cell could eventually become a complete human being. Somewhere inside living organisms existed a set of instructions, a biological blueprint responsible for building every plant, every animal, and every person on Earth. The challenge was finding it. By the late 1940s and early 1950s, attention had turned toward a molecule known as deoxyribonucleic acid, or DNA. Scientists suspected that DNA contained the instructions for life, but suspicion was not enough. In science, evidence is everything. To prove DNA's role, researchers needed to understand its structure. They needed to know what it looked like, how it was organized, and how it could store information. Solving this puzzle became one of the greatest scientific races of the century. Laboratories across Europe and the United States entered the competition. Some researchers viewed it as a purely scientific challenge. Others understood that whoever solved the mystery first would secure a place in history. Among those researchers was Rosalind Franklin, born in London in 1920 into a family that valued education and intellectual achievement. From an early age, Franklin displayed a remarkable curiosity about the world around her. While many children accepted things as they were, she wanted to know why they were that way. Teachers quickly recognized her intelligence, but intelligence alone did not guarantee success. At the time, women faced significant barriers within science. Opportunities existed, but they were limited. Recognition was often harder to earn. Expectations were lower. Many talented women found themselves fighting for positions, funding, and respect in ways their male colleagues never had to consider. Despite these challenges, Franklin excelled. She studied chemistry and developed a reputation for being exceptionally disciplined. Friends and colleagues often described her as serious, direct, and intensely focused. She was not the type of scientist who sought attention. She preferred data to speculation and evidence to popularity. While others might become excited by bold theories, Franklin remained cautious. She wanted proof before drawing conclusions. This trait would become both her greatest strength and, in some ways, a source of frustration during the race for DNA. In 1951, Franklin arrived at King's College, London, where she was assigned to study DNA using a technique known as X-ray crystallography. The method was among the most powerful tools available to scientists at the time. By directing X-rays through a sample and recording how they scattered, researchers could gain clues about the internal arrangement of molecules. The resulting images appeared abstract, almost artistic, to anyone unfamiliar with the technique. Yet hidden within those patterns were extraordinary amounts of information. Interpreting them required patience, experience, and a deep understanding of physics and chemistry. Franklin possessed all three. The environment at King's College, however, was far from ideal. The institution was highly respected, but internal tensions were common. Relationships between researchers were often strained, and misunderstandings emerged almost immediately between Franklin and Maurice Wilkins, another scientist working on DNA. Both were talented. Both were ambitious. Yet communication problems and conflicting expectations created a difficult working relationship. Historians continue to debate exactly how these tensions developed, but there is little doubt that they contributed to some of the conflicts that would later surround the DNA discovery. While Franklin worked in London, another team was pursuing the same goal in Cambridge. The team consisted primarily of two men whose names would eventually become famous around the world: James Watson and Francis Crick. The two scientists could not have been more different in personality. Watson was young, ambitious, and eager to make his mark on science. Crick was older, energetic, and possessed an extraordinary ability to think about complex problems in creative ways. Together, they formed a powerful partnership. Unlike Franklin, who relied heavily on experimental evidence, Watson and Crick focused on theoretical models. They built physical representations of molecules, testing different arrangements until they found structures that matched the available data. Their approach was faster, riskier, and sometimes controversial. As months passed, the race intensified. Every new experiment carried enormous importance. Every clue brought researchers closer to the answer. Franklin spent countless hours refining her methods, improving the quality of DNA samples, and collecting data that surpassed anything previously achieved. She understood that precision mattered. One incorrect assumption could send researchers down the wrong path for years. Rather than rushing toward conclusions, she carefully examined every detail, determined to let the evidence guide her. Then came the breakthrough. In May of 1952, Franklin and her graduate student Raymond Gosling produced an image that would become legendary. Known today as Photo 51, it was one of the clearest X-ray diffraction images of DNA ever obtained. To most people, the photograph looked unremarkable, but to experts, it revealed something extraordinary. The distinctive X-shaped pattern strongly suggested that DNA possessed a helical structure. The image contained critical information about the molecule's dimensions and organization. It was a scientific treasure hidden in plain sight. What happened next remains one of the most debated episodes in modern science. Without Franklin's direct knowledge, Photo 51 was shown to James Watson. When he saw the image, he immediately understood its significance. Combined with additional information available to him and Crick, the photograph helped confirm key elements of their developing model. The race was entering its final stage. In 1953, Watson and Crick published their famous paper describing DNA as a double helix. The model was elegant, revolutionary, and ultimately correct. The scientific world reacted with excitement. Newspapers celebrated the achievement. Researchers recognized its importance immediately. For the first time, humanity possessed a plausible explanation for how genetic information could be stored and copied from one generation to the next. It was one of the greatest breakthroughs of the 20th century. Yet even as the discovery transformed science, questions lingered about the contributions of those involved. Franklin's photographs had provided some of the most important evidence supporting the double helix model. Her calculations had been remarkably accurate. Her work had brought researchers closer to the answer than ever before. Yet public recognition flowed primarily toward Watson, Crick, and Wilkins. The years that followed would bring both triumph and tragedy. While the DNA discovery continued reshaping biology, Franklin moved on to other groundbreaking research. She began studying viruses and made important contributions to understanding their structures. Many scientists believe she was entering the most productive period of her career. Unfortunately, her health was deteriorating. In 1956, she was diagnosed with cancer. Despite undergoing treatment, she continued working, publishing papers, mentoring younger researchers, and pursuing scientific questions with the same determination that had defined her entire life. Rosalind Franklin died on April 16th, 1958. She was only 37 years old. 4 years later, Watson, Crick, and Wilkins received the Nobel Prize for the discovery of DNA's structure. Because Nobel Prizes are not awarded posthumously, Franklin could not be considered. Yet over time, historians and scientists began re-examining the story. As more documents became available, the importance of her contribution became increasingly clear. What had once appeared to be a simple tale of scientific triumph revealed itself to be far more complicated. Today, Rosalind Franklin is remembered not as a forgotten footnote, but as one of the central figures in one of the greatest scientific discoveries ever made. Her story reminds us that history is often more complex than the versions found in textbooks. Great discoveries are rarely the work of a single individual. They emerge from years of effort, collaboration, competition, and sacrifice. And sometimes, the people who contribute the most are not always the ones who receive the recognition first. More than 70 years after Photo 51 was captured, its legacy continues to shape the world. Every genetic test, every breakthrough in biotechnology, every advancement in personalized medicine traces part of its history back to that photograph and the scientist who created it. The image itself is simple. A pattern of light and shadow frozen in time. Yet hidden within those marks was the key to understanding life itself. And behind that image stood Rosalind Franklin, a woman whose dedication to truth helped unlock one of humanity's greatest secrets and whose legacy continues to echo through every corner of modern science.